Hello guys, my name is Dr. Varun and you're watching Chlorophyll classes only on YouTube. This is the last video of the evolution topic. I hope that we have able to complete it in a good way. And if there's any doubt, you can always connect on me on my Instagram, that is at the rate chlorophyll_classes, where your doubts will be taken care with well in the hands. Well, let's just try that what we have left with it. That is the two topics, that is Hardy-Weinberg principle and the what we say that about the human evolution. Before to go with it, the new some extra topics has been given by the CBSE for you as a supplementary materials for your examination, and definitely they're going to be asking you those in your board examination for sure, but it can even go beyond also. So one such topic from evolution which I could remember right now is that about the neo-evolutionary theory, where they have compiled together all the features of the Darwin theory and the mutation theory, which are correct without any criticism, into one such thing, and certain pointers has been added up to you as a new evolutionary theory, which you will let me know that whether you get it or not. Coming back to the major thing, there's a two parts we are dividing this video. One is the Hardy-Weinberg principle and second is the human evolution. Let's just see what it is. Hardy-Weinberg principle, they are the two people, that is G.H. Hardy and the W. Weinberg. They both, these two person, what they did, they give an equation where they tell that if there is any new things are being added, new alleles being added in the population or not, and on that basis whether the evolution is taking place or not. And their equation has certain what we say, the set of principles which they had to follow. If those principles are not being followed, then definitely evolution is taking place. So what are those principles that has to remain constant so that no new gene is added? If any new gene is added, means the new kind of features will be given to the old existing species, and anything new that has been added means what? Means a new species or new characters added up in the species. First, the population must be very large population should be there, so that all the genes, possible type of genes, are present in it. Then migration of individual into and out of the population must not occur, means people from India should not go and migrate to the Europe, and Europe people are not coming to India and interbreeding it, because those people have a different genes. That's why they have a different ethnicity. We have a different genes, we have a different ethnicity. Okay, then mating must be random within the closed population, means that a big population but not going anywhere away, there should be mating properly happening between all. Like that means what? Like India is such a big country, so mating should have happened between, if we see about the humans only, between the Kashmiri and the Kerala person, island person with the northeastern person, vice versa. Whatever is the Indian geographical area, everyone should mate with each other, although it is mating in our country, but on not what? A random way. So most of the genes are in a population, closed by population. Then mutation must not occur. There should not be any mutations. New genes should not be forming, new things should not be coming up and getting the chance to experiencing it. And all genes must have equal chances to pass to the next generation, means no gene should be like that this will go to the next generation or this will not. That should not happen. All the genes should being able to pass on to the next generation. Anything of this is not being followed will lead to what? Evolution. And this is one such question which can be asked to you, and on that basis they give a mathematical equation. So what they say that if there is a population and it has a gene which has two different alleles, okay, for example tall, dwarf, that kind of thing, okay, only two of this is present, okay, so capital A and the small a. So what is the frequency of capital A? It could be P, any number. Then frequency of occurrence of alleles small a will be Q. And if I see P plus Q should be 1, because that is the total amount of population that would be present, that is equals to 100 or 1, whatever be the number we want to take it. Okay, so that is P plus Q should be 100, or anything like P plus Q, if I remove the 100, should be 1. So P plus Q should be 1 only. Coming up to the next, the frequency of the homozygous dominant genotype, means if homozygous is there and dominant, for example A, capital A is taken as a dominant one, if frequency of one allele coming is P another allele coming will be what? It will be multiplied because it's a large population. Now, so we have to multiply. We won't plus it. There are not only two people. It should be multiplied by that number, right? So P plus P, that is P square. Same goes with the recessive Q square. Now, there are the cases where there is a heterozygous condition also present, means P and Q both is present. So how much that P and Q would be present? That will be 2PQ. Okay, so 2PQ is the frequency, and if we take it down completely, that is P square plus Q square plus 2PQ is equals to 1, and that should remain constant. And if it is not coming equal to 1, okay, this thing is not coming equal to 1, means evolution has taken place. So this is the principle. On this basis, lots of numericals will be there that you have to remember how to solve it, which we will be soon doing it. Okay, coming up to the next thing, what are the factors which affects Hardy-Weisman principles? Means how the evolution can occur. We know the principles based on certain postulates, okay, but there are certain factors when at that time this principle won't work. So there are total 5 of them. What are they? We have genetic recombination, gene migration, genetic drift, mutation, and the natural selection. Let's just study one by one. Genetic recombination means new genes are coming up and they are forming the new combinations. How could that even possible? Let's just revisit back to our whatever we have studied in our previous biology lectures or in our school understanding. So number one is at the time of the crossing over during mitosis, not mitosis, meiosis. So means when your cells are undergoing what we say that cell divisions during the time of formation of the gametes, you know that there is a prophase 1. In the prophase 1, there are certain steps. There is a one such step where crossing over occurs between the non-sister chromatids. You might have studied that. If not, that go back, check it out, talk with your teachers. You can connect with us also on the chlorophyll classes on our Instagram page. We will try to help you out with the concept. Coming back, so during the meiosis, crossing over occur, and if the crossing over is occur, means new things are being added up. That's how the variation comes up, right? Then we have what? We have independent assortment that occurs at the time of gamete formation. We know that new recombinants are being formed. That's why in the genetics, if you're not seen that, go back and check my genetics video on this channel, and you will see that certain genes, they are able to do what, like they come into the recombinating form. Independent assortment takes place in their cases. How it takes place, I have explained it much better. Then the random fusion of gametes during the fertilization. So any gamete can fuse with anyone, and you know that gametes are different gene compositions, and it could lead to more different recombinations or new varieties of the variations, and this causes what? A reshuffling of the genes and the alleles in the population, which leads to what? Which leads to change of the addition of new alleles or different alleles into the population, and it diverts what? Your Hardy-Weinberg principles, because earlier there was this and this allele. What if this allele also come up? A dash, right? So this thing is there. Then comes the gene migration. Very easy. It means what? That people from one area goes to another area. We already know that migration should be 0 because new areas will have a different kind of alleles present. Okay, different varieties would be present over there. So here what happens, if a person moves from India to US and the US to India, anyone vice versa, and over there they mate with the people over there, then they are adding up the new genes into the population, and it's deviating the whole situation. Now what is a gene pool? It is a total collection of all the genes present in a population, in a closed population. Okay, means India, China, closed country. Understand? Then gene flow is what? Exchange of gene of two different population of a species is called as gene flow. For example, one population is of Europe, other population is of China, and they are mating. So they are doing what? They are doing the gene flows. Okay. Then comes the genetic drift. What is genetic drifts? It is called as Sewell-Wright effects. Okay. So Sewell-Wright effects, he said that any gene frequency can change. It could be a chance alone feature or by natural selection, but just like in a fraction of moment. Okay, so that leads to what? A genetic drift, a change in the genetic pool and the constituents and the alleles which are present. And it is usually marked in a very small isolated population which has very less organisms, and anything will happen, they will lose their genetic information, and it leads to what? Reduction in the genetic variability. And it could, like, if certain drift happened means certain problem occur and most of the population wiped out. Just think that if the humans are there, and imagine that a huge apocalyptic meteor just ran over us, okay, on the planet Earth, and only 100 humans survived, rest all died. So those 100 humans, do you think that they will have all the different types of alleles which are required? Because more the alleles, more with the survival opportunity. Because you know that when we are studying the mutation, we will understand that thing more better, that certain times when we have a mutation, like you have studied natural selection in case of DDT mosquitoes, right? We said what? We said that DDT mosquitoes are able to survive because they have that kind of mutation present, means those genes present which can survive, but they are not able to show their effect because DDT environment was not there. We are not splitting the poison on the mosquitoes, right? Now here what happened? Only the 100 humans are left. Now these 100 humans, let's say that one such feature is let's say that cold environment adaptability, okay, which is, just think, like, it's not like that, just giving an example that all the 100 of them don't have that, but other people had it, so we might not be able to survive in the cold environment. We have lost that gene. Unless it's a mutation happens in the gene during the time of evolution, we might get it back, but at present we lost it. Okay, so there is a reduction in the gene variability and fixation of the new mutation also, since a small group of organisms are left. Okay, so 100 people have some new mutation, they will fix it permanently, which could be advantages, disadvantages. That is a talk of the town. Okay, then coming up, there are two things we will understand. One is the founder effects, one is the bottleneck effect. What is the founder effect? Simple meaning, for example, in isolated area we humans are living, right? And imagine that the human population is only 100. Okay, 100 is in the whole world. Okay, so 100 humans are living. Soon a flood came. Okay, and what happened? 90 of them died, only 10 left. So those 10 people will repopulate themselves, they will mate and they will repopulate their species into next number. So what we have understood with this, the people, the 10 people which are left, whatever the genes they are, they becomes what? They becomes the only source of information for that species, and they become the founder of the new population of the species. This is what they are telling you over here. Next is bottleneck effect. Bottleneck effect, for example, an organism is living in the population and the population is totally exploded. We take those people from there and we give it into a new population. Over there, they survived. They also loses someone because of the calamities and all the problems that are coming up, natural problems that are coming up. And whatever is left, they grow and they becomes a totally different species than the original one. How we will remember it? It is very common to see in the bacteria cases, not in the human cases, but I am giving an example that, for example, humans are there, few of them, since the planet Earth is fully occupied with us. Now we don't want, we want a new home. Okay, so we took few people from planet Earth and put them on the Mars. And on the Mars, they were able to survive after millions of centuries. Okay, when we see us and them, they will be very different because in the Mars environment they are adapted and all those things. They might be living without the oxygen over there or the low quantity of the oxygen over there. So I hope this thing is understood, that what is the bottleneck effect, that certain population, we take it from here, go to the new places, they becomes a new species, a totally different species, which can happen. Then come the next, fourth one, that is a mutation. Now understand the mutation. Okay, mutation means that even a single thing, okay, if you give like DDT environment, if you remember that the same thing is there, natural selection wala thing. Okay, so for example, here what happened, there was a leadwork replica plate experiment. They did an experiment on certain bacteria. They did what? They take a bacteria in the Petra dish and growed them. Then they imprint the same thing on this. Okay, and after imprinting, they imprint the same thing on the new thing. Now why they did it? So that they can make two different colonies. If you remember where we are doing this, we have done this in the biotech also, when we are making the antibiotic resistance criteria as a selectable marker. So what we are doing, just imagine that, for example, this is the wood kind of thing. Of course, it will be much bigger than that. We put a velvet over here. Okay, I don't have anything. Okay, I will get that something. Imagine that this is the cloth. So imagine that this is the velvet cloth. I put it on this wooden block. Okay, so and I have made a plate over there, and now I have to make a imprint of it. Why I have to make it the imprint of it? So that I can study. Okay, so that I can correlate. Now understand this. This is the first plate. Okay, I have grown the bacteria. Okay, and then I imprint this. For example, this should be bigger, same size. Okay, imprint this and put it in the new. There is another plate. I put it on this and gently press it. Not do it like this. We will make every bacteria killed. Okay, so we will just gently put it on the new plate with our medium of the growth. Okay, now this and this both will have what? Similar area where the similar kind of bacteria would be there. So for example, this green color bacteria will be green color bacteria. This colony would be red color. This colony would be red color. So we are seeing what? We are seeing that similar imprint is there. Now what I did, this is my master plate. Okay, because main plate is there, because I will correlate with this, the first one. The second one is what? Replica plate. Okay. Now I add what? I add antibody. For example, penicillin. Now penicillin is antibody. Antibody means it will kill the bacteria, because I have made the bacteria only. It killed a lot of bacteria, but certain bacteria still survived. Okay, rest of the bacteria killed. I came to know what? One thing knows that when I put the antibiotic resistant penicillin, when I added the antibiotic penicillin, then those colonies which will be killed up, means those bacteria that will be killed up, I can easily see over here, okay, that this is being killed, right? This is how we use that. Okay, second thing also I can learn that certain bacteria which do not kill, because I added the penicillin on it, those do not killed up. They are what? They are the resistant ones. Means they have the particular feature. What is the feature? The feature of resisting the death. Or why they are resisting the death? Because they are what? They have resistance gene present, mutation present already, but it's a chance factor that they had been exposed to the environment which helped them to come up their superpowers. Okay, like the Stranger Things that not gonna give any bloopers, but there's a person who find out his hidden powers when the situation came up. That is also what he got certain mutations. Okay, let's coming up back to the normal. That is, this whole thing has been showed to you that this is the bacteria. This bacteria over here, during the course of its life, they are evolving with the time, right? One bacteria got a mutation of what? Resistance to the penicillin. So once we put the penicillin on this generation, this is the first generation, second generation, third generation, fourth generation, they all died because they were not having any resistance capability, right? They have the resistance capability. Resistance to what? Penicillin. They survive and they grow and they maintain the bacterial population with a new different genes. So mutation helps like that way. Okay, so mutations are not that it will be soon as they will be present and we will be able to find its effects. It's a nature that provides or select on the basis of that. All the example of DDT, industrial mannerism and all works over here as well. I hope you understand the mutation. And we come on to the last one. That is what? That is the natural selection. In natural selection, what has happened? Nature is selecting something over the other. Totally nature's own way of finding the who is fittest. Okay, fittest. Okay, not fetish, fittest. So if we see that, if this is for the height, let's take it. We know that most of the people are what? Mediocre heights. Okay, human population, right? So more people would be what in this? Like for example, this is 1 feet. 2 feet, 3 feet, 4 feet, 5 feet, 6 feet, 7 feet, 8 feet, 9 feet, 10 feet. Let's take it like 1 to 7 feet. 7 feet are very less, 1 feet are very less. If we go over here, this is what 2 feet, this is 3 feet. Okay, then 4 feet to, let's say that 5.9 feet. A lot of people are there in this range. Average 5.5 maybe, or 5.4, right? So these people are more in height. The mean value people are more in height, right? On that basis, like this is what we have said it, that people are in the different heights. And the last example which I told you, that is becomes what? A stabilizing selection. Means nature is selecting what? Those people which are of mean height. Mean means center. Okay, not too big, not too less. Okay, so here you remember that. So what will happen? This whole thing earlier was like this, but these people will not be selected. So do you see anyone of 1, 2, 3 feet? Hardly no, right? Do you see anyone of height 7, 8, 10? No, very less, not at all. So what happened? This whole curve comes towards the more peaker height. Height is increased of the graph, not the person, right? Height of the graph increases, plus it becomes narrower because the extremes, extreme small, extreme tall people are not in the population. One or two can form, but mostly are not. So we see that most of the people are coming up in this height range. So this is being told. Now what if nature start selecting any one extreme, maybe very small heighted people or a tall heighted people? So what will happen? This whole thing, the people won't be present here at all if they are selecting the tall heighted. So most of the people will be here. So the graph will become what? Comes towards this side if more people are present on this heighted, right? So the peak shift to one direction. And what if nature stop preferring the median or the mean value people, they're only supporting the extremes like short heighted, tall heighted. So what happened? The short people will be more, tall people will be more, medium will be very less. So you can see that dip is there in the center. So means this height, means the mean height people are less. So this becomes what? This becomes disruptive. Disruptive selection. Okay, I hope this thing is understood. You will be asked in your board exams to make the diagram. They will give you certain options. I give you with the example. They can go with anything else, and then you have to find out whether it's a stabilizing or which graph will be forming up. Coming up to the next thing, one thing that is there in your syllabus, but not we not asked. Okay, so what is that? That is the balancing selection. What does balancing selection means? It means that certain alleles which are in the equilibrium given into the place, for example sickle cell anemia, how it is balancing one? If both this this is given, this will be what? Fatal, because the person will be what? Person will be making only the sickle shaped RBCs, means they are not even able to carry the enough oxygen in their body. Okay, so they can't survive. But the people who have HbA and HbS, means normal also and as well as sickle also, these people survive because normal thing is able to make them compensate for the loss of the other chain, that is other allele which is doing it, right? But HbS is making a little bit of what? They are making the sickle cell where the malarial parasite is not able to stain over there. Okay, because malarial parasite need what? It need a proper RBCs to go inside it and infect. Okay, in its erythrocytic zygogony cycle, which is what we will see later in the human health and disease chapter when we will be studying the malaria. So this is what advantages also. So this is what balancing selection. So nature said that okay, you can keep this allele is good in certain case, but it becomes harmful when two carrier comes up and marry up. So it's very important. The fact which we will suggest, if you're getting married up, go check for these kind of genetic defects or your markers for the sickle cell anemia and all those things. This is a common disease that we encounter. I hope this thing is understood. Balancing selections. With this, we understood Hardy-Weinberg principle. If there's any doubt, let me know in the comment section. Okay, coming up to the next part of this video, that is a human evolution. Let's make it clear. Okay, human evolution, if you know that what is happening, you can easily understand that what's gonna happen. We're gonna see that how the human comes up and where it comes from. Of course, we have a linkage with apes, but we came from the apes is something wrong. We came from ape-like ancestor. Means apes and humans, they become two different branchings, means branching descents. One form the apes, other forms the current humans, right? Let's see that how that happens. And that becomes our topic of interest today, that is human evolution. So if you see this whole thing, this is the different epochs. What are the epochs? Let me know in the comment section if you know what is the epochs. So they were tree shrews. These are what? These are the initial mammals. They forms what? Lemurs and all those things. And from them forms what? Two different varieties form. One is the old world monkeys. Okay. Another is the Parapithecus. They form the old world monkeys, new world monkeys, and Dryopithecus. Now, here the story comes up. Dryopithecus. It is considered to be the most similar, okay, not very similar, but a too much similar lineage, okay, where we can see that a new species is being coming up. That is Dryopithecus. Dryopithecus give rise to this gibbons, organton, chimpanzees, means these are apes variety, as well as it gives to Ramapithecus. This thing is going where? Going towards Homo sapiens like us. Means Dryopithecus gave apes as well as human lineage, right? So since we don't have to see that how the apes are being, the chimpanzees and all those things are formed, we are more interested towards the human. Let's just see that what is happening with the human. If we see that the Dryopithecus gave Ramapithecus, Ramapithecus has a lineage with the Australopithecus. Then Australopithecus gave very varieties which end abruptly. They died, okay? But they give one other lineage, that is Homo habilis. From Homo habilis comes Homo erectus. Homo erectus gives two different lineage. One is Neanderthal men's, very intelligent, but they also died. Then Cro-Magnon man, but they also changed into Homo sapiens. NCERT do not have this Cro-Magnon man. NCERT says that it's a Neanderthal men's and the Homo sapiens. So we will remember like that. Now the major question is how I will remember this whole big thing, that Dryopithecus, Ramapithecus, Australopithecus, Homo erectus, Homo habilis, Homo erectus, Cro-Magnon or Neanderthals, and the Homo sapiens. You are a teacher, you have to mug it up. But how would I remember it as a student? So there is a one trick for it. And the trick is, that is, where is that? The trick goes something like this. That is Dr. Sneha. What is Dr. Sneha? Dr. Right? Sneha. Now we will understand it. Now here you will, as you have right Dr. Sneha, so you will start from here. D for Dryopithecus. So from here everything started. For R for Ramapithecus. At least remember the name. Dryo is there, Rama is there, Australia is there, right? Ramapithecus. Now what happened? A dot came. Means full stop. Stop. So you will like, okay, I will go towards the other side. So other side is this. This way you have to run now again. Now first is this, second is this, third is this. So A for what? A for Australopithecus. Then H for, this is Homo habilis. Actually they have taken only for the habilis, right? Homo word is coming up again, right? Then E for is what? It is Homo erectus. So we have taken for E, that is erectus, that is fourth, fifth. Then comes this sixth N for Neanderthals and S for Sapiens, which is the Homo sapiens, the latest one. I hope you would have understood this thing, how to remember this thing, right? So let's see that from where we have started. So we have divided into three parts prior to the ape man, before the ape-like structure, then ape and man similarity kind of fossils, and then we see the modern man-like fossils. The first one is Dryopithecus. We found it in the Africa and in the Europe, and they existed around 15 million years ago, the most oldest form. Now, it is a common ancestor between apes and mans. We have already seen in the diagram of the lineage, right? They give this thing, apes, and they give the man-like characters like Ramapithecus also. It is more like ape, looks like the chimpanzees and all those things. Arms and legs are of same length. If you see us, where arms and legs are of not same length. Okay, they have what? They have large canines, incisors are there, and they feed on the fruits and the leaves. They are not animal eater. Okay, they are they are knuckle walker. They walk like apes, right? So this thing is there. Then comes Shiva Pathicus. The name itself says something like Shiva. Shiva means India. Yeah, it is seen in India, where in the Shivalik hills. What are Shivalik hills? These are one of the range of the Himalayas. Himalayas are actually three different ranges, okay, mountains in the line, right? So Shivalik hills are one such range of the inner ones. Like outer one is towards the China, inner one is towards the Shivalik, okay. So in those hills, we found Shiva Pythecus, which looks like Dryopithecus only. Okay, so it has monkey-like and the ape-like structure present. But yeah, of course, we can see that it is what Dryopithecus similarity. Similarly, we got the Ramapithecus. Rama word is also from the Indian mythological thing, and that is what from Shivalik hills only. In the Pliocene epoch, we found what? Another fossil around 14 to 15 million years. More, it arises from the Dryopithecus. We can see that it's much different. And in a 1 million, there's a change, a lot more change that happened. It's more like man, teeth and jaw is more like humans. Before this, it is what? Jaw and teeth like what? Like a ape. Let's come to ape man, including prehistoric man. So here we come up with more varieties, three varieties here again we're going to be studying. Number one is Australopithecus. It is called as, the full name is Australopithecus africans because it was seen in case of Africa. And in the Tuang, Africa, we found it. The first skull we found it is of 5 to 6 year old baby called Tuang baby around 2 million years ago. Okay, that's a lot of time has taken place. That's why there is a lot of difference also seen, because earlier we were studying about what? 13 to 15 million. Here we are talking about 2 million. So means almost what? A time frame of 10 to 11 million years has occurred. So of course they will look more like men. So that ape men, including prehistoric men, they hunted with the stone weapons but ate fruit. So why they hunt? They hunt for the protection. Okay, so they are both man and ape like bipedal motion. So they can walk upright also on their feet, right? They are omnivorous, erect posture. Teeth were man-like. Brain, of course, man-like teeth has already been formed, jaws and teeth. So here they will be like that. Brain with the ape-like cranial capacity means its mind capability is what? It is 500 cc, which is same like ape. Okay, then lifts in the caves, bow ridge is present, this ridge over here present. Vertebral column has a lumbar curve, only one curve present over there. Now they have different varieties like Africans, Afrans, Robusta, Boys, Ramidus and Ethiopus. All of them died. Okay, nothing survived. Every of this died, but the Robustus has 600 cc of the cranial capacity. Cranial capacity is very important for you to remember. It is increasing, little bit decrease with us. Okay, that we will learn it. Coming up to the next one, that is Homo habilis. We found this in the East Africa. They are the first hominid human-like. That's why Homo genus is given to them. They are the tool maker. That's why also called as handyman, because they were buried with their tools. Okay. and they were made up of stones. Cranial capacity is increased 650 to 800 cc. Before this, we have seen that African robustus has what 600 cc of the cranial nerve. It leads a community life, means lives in a community like we live in the community now, in a proper human community, like this is Muslims and all those things, or maybe in the society, gated society, that's the thing. Then cared for the young ones like us. Then probably do not eat meat. Teeth were human-like, goes like that. Then Homo erectus, we found it about 1.7 million years ago, means more recent kind of fossil we got it. Cranial capacity is increasing, it becomes 900 cc. Before that it becomes what 850 cc only. Hunted large animals, perhaps know the use of fire, and it has three fossils. One is Java, we got it from the Java, Indonesia, Central Java. Then Peking Manx from the Peking in the China, Heidelbergs in the Germany. Okay, and their cranial capacity is what 9, like if you see the Java man, the cranial capacity is 950. If you see the Peking man, it is 850 to 1100, and the Heidelberg is the 1300 cc. A lot of variations in the cranial capacity is there. So, but one important thing is that they all are, mostly of them are what, they are cannibalism. Cannibalism is the brutal form of the species interactions for the existence. If you learned about that in the ecology, and we have learned something about that in the Darwin existence for struggle for the existence, where cannibalism leads to what, proper species destruction. And they are cannibal means they ate similar people to themselves, means that becomes what, a major problem for the species to survive. Okay, so this thing is there. Then comes the true men, including modern men. We have Neanderthals, we have Cro-Magnon men. Neanderthals, we found it in the Neanderthal Valley, and they walk upright. They are skilled hunters. They used to wear skin of the other animal as a fashion to protect them from the harsh climate, because Germany is super cold. Cranial capacity is increased to 1300 to 1600 cc. They are also cannibal, probably the reason they would have died. They had even a religion also, and they do not evolve into modern men. Okay, but Cro-Magnon men, they have cranial capacity of 1650 cc. It is even increased. They are the direct ancestor of modern humans. It is believed that they have done the cave paintings. They run faster, walk faster, and they're intelligent and the cultured men of the today. Okay, means even the real men, they are more, because the cranial capacity is 1650. Because when we come up to the modern men, the cranial capacity is an average 1450, that is 1300 to 1600 cc. But Cro-Magnon has 1650, so we have significantly decrement in the cranial capacity. So if the Cro-Magnon man is given to you in your answer, you have to always remember that the Cro-Magnon man has more cranial capacity than the current men. So are we any less of them? Maybe we are good for that. Okay, so let's see that they appear around 2500 years ago, and after that different periods comes, like iron period, bronze period, gold period, silver period, like these comes. Morphological transition, there is a slightly raising of the skull. Our skull is raised up a little bit. Thinning of the skull bones. Slightly reduction in the cranial capacity. Development of the four curves in the vertebral column. So we have a curve like this. Okay, cervical curve, thoracic curve, lumbar curve. All of these things. Then morphologically changes. We attained a postures, erect posture, four curves are there, increase in the height, increase in the cranial capacity. If we remove the Cro-Magnon men, then we have comparatively increased our cranial capacity only. Well, that's all about the human evolution. You have to remember strictly what is given in NCERT. Here I have taught you so that you can feel the vibe of this chapter, that if these things comes up, right? With this chapter, that is evolution, is finished. If you like this video, let me know in the comment section. Like, share, and don't forget to subscribe. We are coming up with more things on chlorophyll classes. Thank you.